Okay, so hello everyone, and thank you so much for joining us at Biologically Speaking's last webinar of 2021. This really has been a great year for us, first with the success of our Sunday webinars, but also with our two flagship conferences, Her Story of Science, which celebrated the contributions of women researchers, and then Simplify, our science fest for all the young science enthusiasts out there. And we also feel extremely privileged to have been sponsored by BiosWatch India and the American Society of Cell Biology for our science outreach efforts. And a big part of this success is you, the audience, who have stuck by us through all our endeavors. And we hope to keep this association going in the coming years as well. Today, we are joined by none other than Professor Oded Rechavi from the Sagol School of Neuroscience at Tel Aviv University in Israel, whose vast body of work and range of interests has been a delight to read about. But today will be even better because we get to speak to him in person. Professor Rechavi has obtained both his bachelor's in neuroscience and his PhD from Tel Aviv University in Israel, following which he did his postdoctoral research at the Howard Hughes Medical Institute in New York in the US. He has been at Tel Aviv since 2012, again in varying capacities, and now is a full professor there. Professor Rechavi's research interest primarily revolves around the C. elegans biology. And as his webpage says, Professor Rechavi finds thrill in challenging the long-established dogmas in biology. And today he will tell us some more about all of this himself. So a warm welcome to you, Professor Rechavi, to this platform, and the virtual platform is all yours. Thank you.
Thanks a lot for the kind introduction and also for the invitation. It's a real pleasure, and it's a great initiative that you have there. And so I was thinking, what should I talk about today? And together with the team, I decided to try to improvise a little bit, and instead of presenting a PowerPoint with a million different slides, usually I bombard the audience with a lot of data. I decided to instead try to be a little spontaneous and just talk with you without slides. And let's see how it goes. I thought that since we're already 2 years into this pandemic and so many Zoom talks, perhaps it would be refreshing to try to do this a little differently and not use the presentation. And perhaps like this, I can tell you about different things, and also, just follow your questions or your ideas in case you want to intervene in the middle. That's perfectly okay. You can ask a question and I'll respond gladly, or develop the topic according to your ideas. So in my lab, we studied many different things. We studied epigenetic inheritance in C. elegans. We studied additional questions as well that are totally unrelated to C. elegans, like the genetics of the Dead Sea Scrolls and irrationality, all kinds of things. But the main topic that interests me always is memory. I've always been interested in how memory is encoded. In fact, this is why I went to study C. elegans, because when I was studying neuroscience, it's the bachelor degrees, I realized that we don't know enough about memory, and it's extremely hard to understand and study because our brains are so incredibly complex. So I wanted to study it in the simplest organism, or in the organism with the simplest brain. And C. elegans nematodes have a brain, or a nervous system, more accurately, that is comprised of just 302 neurons. That's a very small number in comparison to mice or flies or, of course, humans. And for these 302 neurons, we know a lot about them. We know each neuron by its name and to which other neurons it is connected. So we have a full connectome of the brain of the one, and it's a very powerful tool to study how the nervous system works, and specifically how memory is encoded. The problem is that C. elegans is, of course, limited in the types of memories that it can learn and keep. And this is something that I will get to later on in this talk today. Okay? That's a big problem. Of course, if C. elegans could do the things that we do with using its brain, it would be amazing, but it can't. And one of the
I think there's some technical difficulty. Please bear with us. I'll try to see.
I'm back here. I was cut off. Can you hear me now?
Yes, we can hear you. Perfect.
Okay. So should I continue?
Yeah, I think your video is off, but
oh, sorry.
Other than that, it's
yeah,
fine.
I see. I mean, it's on my computer. Perhaps it's turned off at your computer, or
can
you see
does
it now?
Everyone else? No. Is it
No?
just me, or can no one see the video? Did someone
no,
just write in the? Okay.
No, but no, because the video is on my computer. So perhaps it's just everybody's stream is, everyone is muted or something like this. Could it be?
I think Oded, there's some connectivity issues with your internet. Can you check once?
Let me. So I'm open.
Yeah, but we still cannot see your video, unfortunately. Would you mind rejoining, maybe, if that works? Yeah, so sorry for the inconvenience. Please bear with us. I don't even see like the icon next to you. So that's, I don't know what the problem is.
Can you see me now?
No. Yes, now we can. Yes.
Now you can see me?
Yes.
Okay.
Perfect.
Okay, so let's hope everything is working from now on.
Yeah.
And I will just continue, okay?
Yes, perfect.
Okay, excellent. So the last thing I was saying, I believe, was that C. elegans has a very simple nervous system comprised of just 302 neurons. It can learn certain things, but of course it has its limitations. It can't learn, do the things that our brain does. But nevertheless, it can do quite complicated things. For example, the worm can sense thousands of different odors, can do chemotaxis, can move towards these odors, can sense light touch or harsh touch, can move in complicated ways, can reproduce and mate, and even is capable of simple forms of social behavior. For example, the worms can aggregate together with other worms when you increase the concentration of oxygen to lower the concentration locally. So it's quite sophisticated, although it has a brain of just 302 neurons. And very importantly, the worms can learn simple associations. For example, you can teach them that a certain odor that they like shouldn't be liked if you pair it with something bad like starvation. Okay, so I thought that I'll go and study memory using these nematodes, which have a very, very simple brain. And the thing about memories, we know that they are complex and dynamic and hard to study. And the angle that I was going for is that's the idea that I had, and I tried to explain it, is that we knew that I thought that if some memories can transmit across generations, then perhaps at one point they will exist at molecular form. What do I mean? At some point, these memories will be molecular. What do I mean by this weird saying? If a memory is transmitted transgenerationally, it means that it has to exist at some point inside a single cell, because we are all formed from a single cell fertilized egg, which at that point, when it's just this cell, doesn't have any synapses with other cells. It's just this cell alone. So if a memory transmits between generations, it means that at some point in time, it has to exist as a molecule inside this single cell. And perhaps during this stage, we can characterize it molecular, just like we sequence DNA or RNA and understand and have a grasp of this memory. This could be a sort of a memory in a tube. This was the idea that I had very naively in the beginning. Now, we know on the one hand, we are not born, that some memories are held in synapses. That's very clear, but these are sophisticated memories. They require reshaping of neuronal circuits. On the other hand, we know that we are not born blank slate. Some memories do exist, are inherited transgenerationally, and therefore do exist as a molecule, perhaps encoded in the DNA or encoded in another form, because we are not born as a blank slate. We are born with some information encoded already in the hereditary material. For example, experiments that were done in the 80s in the US have shown that monkeys who are born in captivity have an innate have a capacity to learn to be afraid of snakes, but not flowers. What do I mean? So they've shown that these monkeys, which are born in the lab, they don't know anything about aside from what the experimenters teach them. If you try to teach them to be afraid of a toy flower, you present them with a toy flower and couple it with something scary like an electric shock or a loud noise, the monkeys will have a very hard time learning that be afraid of this toy flower. You'll need to pair the toy flower to the electric shock for multiple times until the monkeys learn that they should be afraid of toy flowers. They will eventually learn, but it will take them a long time. On the other hand, if you try to teach them that a toy monkey is scary, by pairing the toys, if you try to teach them that a toy snake is scary, by pairing a toy snake to something like an electric shock or loud noise, they will very easily learn this association and be afraid of the toy snake. They are not born afraid of toy snakes, but they can easily learn it. So this means that they somewhere in the hereditary materials, there's information telling them that snakes are scary and they should learn to be afraid of snakes quickly, but flowers are less scary. The question is, where is this information? Is it in the DNA? Is it in some other molecule? And how do we study it? It's quite hard to study this in monkeys for many reasons. It's also not something that I would personally like to do, but it's much easier to do it in C. elegans because of the simple brain, the simple nervous system, and also because you can very easily separate nature from nurture in C. elegans and grow them in very defined conditions. Normally, we grow them in a plate with just agar and food, and the food in the lab is E. coli bacteria, and you can control their temperature and all of the environment. And in addition, the worms are almost genetically identical to their children. They are not clones, but since they make both sperm and egg and can self-fertilize, they have very little genetic variability. So you can distinguish between what they learn and what they are born with, and see whether indeed memories are inherited across generations. If so, have a relatively good chance to understand how it is inherited and where does the memory sit, where is it encoded. And that was the idea that I had when I started working on C. elegans. I only started working with C. elegans in my postdoc. Before this, I did something else. Now, the problem is, of course, that the concept of inheritance of memories is linked to the concept of inheritance of acquired traits, which is total heresy for biologists. This is not something that should happen. It goes against some of the most fundamental dogmas that we have, some of our deepest beliefs as biologists. We know that memories shouldn't be inherited and inherited traits shouldn't be inherited. So since we know that Lamarckian inheritance is not true, right? This is something that they teach us in high school. However, it turns out that many of our assumptions are not true. First of all, if we go back to the history it's not clear that Lamarck was really as Lamarckian as people think, that he himself believed in inheritance of acquired traits. He believed in what everyone else in his generation believed in. And in fact, Darwin was more Lamarckian than Lamarck. Darwin believed in inheritance of acquired traits. It's even in the origin of the species. He described this theory, which is essentially Lamarckian. But that's a different story. The thing is that we should separate Lamarckian inheritance, or what we think about Lamarckian inheritance, from the idea of transgenerational transmission of parental responses, which is much more limited. It's a smaller concept. It doesn't involve will as the driving force for evolution. And this is something that we can perhaps deal with and study experimentally. And what prevents inheritance of acquired traits from happening is that there are a few factors. One of the main factors is this idea known as the Weizmann barrier, the separation between the soma and the germline. And the nervous system is, of course, part of the soma. According to the Weizmann barrier, this is also known as the second law of biology, only germ cells can transmit information across generations. What happens in the soma, or what happens in the nervous system, in the brain, stays in the soma. It doesn't get passed to the next generation. And of course, we know this from our experiences. If we go to the gym and build big muscles, our kids won't be stronger as a result. Only changes that occur in the germ cells affect the next generation because the germ cells are made from the combination of the sperm and the egg. And only the genetic material of the sperm and the egg affects the next generations. So, and because of this very simple and intuitive concept, acquired traits and also memories that are acquired by the parents shouldn't transmit to the next generation. In C. elegans, however, we know experimentally for many, many years that the Weizmann barrier is breached and that some parental responses that originate in the soma do transmit to the next generation and affect the physiology of the next generations. When Fire and Mello described the mechanism of RNAi, when they've shown the double-strand RNA is a substrate for RNAi, they've shown that for RNA interference, to those who are not molecular biologists in the audience, they've shown that if you inject somatic tissues of C. elegans with double-strand RNA, or if you just feed the worm with double-strand RNA, the silencing response that this double-strand RNA initiates transmits between the different cells of the worm, moves between different tissues, and also reaches the germline and affects the cells of the next generation. So this is a clear violation of Weizmann's rule. The RNAi moves from the soma to the germline, and silencing responses transmit across generation in clear violation of Weizmann rule. This is because we know now the RNA itself can shuttle between different cells. It is not entirely clear which RNAs do this. We know that exogenous artificial double-strand RNA can move between cells, and there are transporters that shuttle double-strand RNAs between different cells, including from the soma to the germline. When it comes to endogenous natural small RNAs, we don't know exactly which RNAs do this, which RNAs are capable of moving between cells. But it is clear that RNAi responses are carried from the soma to the germline and across generations. And I thought when I started working with the worm, this was in 2010, that perhaps such RNA would be capable of transmitting parental responses across generations. People have shown over the years that artificial double-strand RNA induced silencing responses can move between generations, but it wasn't clear whether natural responses or things that happen in the environment can similarly affect the next generation via RNA. I'm just reading a question from the comments. Are there distinct transgenerational inheritance processes between consciousness and unconscious memories? Baby learn how to digest foods by unconscious, when it's born by unconsciously, but do not speak consciously. So in C. elegans, I'm not sure how we can distinguish conscious from unconscious responses, but I'll try to get to this later on, okay? So Mirazulu asked this question, I will try to relate to it later on. Okay, so as I was saying, the starting point of my project was that, for my research project that until today, was that I knew that in C. elegans, RNA responses can transmit between generations, but it wasn't clear at the time which type of environments or which type of responses can transmit to the next generation via RNA, if any of these responses indeed can do it. It wasn't clear at all. And I was looking for a response that might be transmitted across generations via RNA. And the first response that I was studying is an antiviral response. C. elegans are remarkably resistant to viruses, although they don't have dedicated immune cells like we do, like B cells and T cells and NK cells. They don't have these cells. What they do is they use RNA to destroy viruses. And what I've shown, and I won't get into this now, is that the worm can produce transgenerational RNA, transgenerational antiviral responses by shuttling antiviral small RNAs across generations. And this was the first demonstration that a trait can be carried across generations in C. elegans via small RNAs. And we also described some of the mechanisms involved. This is a paper published that I published in my postdoc in Cell at 2011, and I won't get into the details. There are many details, but you can look it up. It was published in 2011. And later, I wanted to know whether there are additional responses that are similarly carried transgenerationally by small RNAs. And the first response that I studied was a response to starvation. The reason that I wanted to study starvation was that the biggest epidemiological study in humans about epigenetics was a study where they examined the descendants of people that experienced starvation during the Second World War in the Netherlands. In the Second World War, the Nazis sieged large parts of the Netherlands, and people studied the descendants of the individuals who were starved. And they saw that one generation and even two generations after the starvation, the progeny of these people showed certain effects that couldn't be explained just based on regular genetics. So they were more, they developed more diabetes, and they also experienced certain neurological diseases such as schizophrenia. So this is a classic example for a parental response that is carried over transgenerationally. However, it's not clear that it's a real transgenerational response, because the people who were starved, the women who were starved while they were carrying their babies in utero. So you could say that the babies were directly affected by the starvation. They were directly starved also. And since these babies in utero also had germ cells, even another generation down the road was directly exposed to the starvation. So it's very interesting, but it doesn't necessitate any new mechanism of inheritance. Only if you go for additional generations and examine babies that are not directly that were not directly affected by the starvation themselves, then you can really talk about a heritable response, and would have to think of a mechanism that can allow it. We studied this in C. elegans, but we went directly to examine unexposed individuals to see whether they are affected by the life history of their parents. And we saw that when you starve worms, 3 generations down the road, you still see an effect. It affects their nutritional state, their lifespan, and people who studied this later also showed that the descendants of starved individuals, even 3 generations down the road, have certain effects. For example, they are more resistant for additional rounds of starvation, and their developmental rate or their speed of development is altered. And this was again mediated by heritable small RNAs, and did not happen in mutants that can't inherit small RNAs. Now we know of genes that are required specifically for transgenerational inheritance of RNA, but not for RNA per se within the same generations. For example, there are Argonaut proteins. These are proteins that carry small RNAs, which are needed just for inheritance of RNA, but not for RNA within the same generation. There are additional genes that have the same phenotype as well. We wanted to study more about the more mechanism, and we studied it. We studied how the duration of the response is dictated. Why does it last for 3 or 5 generations and then it stops? And we identified a feedback mechanism that controls the duration of the inheritance, which is composed of small RNAs that are inherited and target the RNA machinery itself. And if you disrupt this feedback mechanism, you get responses that are very long lasting, that can even last tens of generations. For example, there's a methyltransferase called MET2, and we found that mutants that are defective in this methyltransferase, when they are exposed to RNAi against germline genes, they inherit the responses for tens of generations, just stable, because the feedback mechanism that shuts the inheritance off doesn't work. We studied different components of the inheritance mechanism, identifying genes that function in the nucleus, such as genes that are involved in chromatin modifications, and also genes that function in the cytoplasm. Recently, we've been studying liquid-like germ granules that reside just outside of the nuclear pore, where many of the components that are required for RNA inheritance reside. And these granules sort the small RNAs that will be inherited transgenerationally. And if you disrupt the granules, you get long-lasting effects that last for tens of generations. Now, going back to the original ideas about memory, we found that different tissues in the worm can generate transgenerational responses. For example, if you feed the worms with double-strand RNA, the response starts in the gut, where the RNA is digested and then moves to the next generation. But additional tissues can also generate transgenerational responses. And the most interesting tissue, in my opinion, is the nervous system, because if the nervous system can control inheritance and can initiate small RNA-based transgenerational responses, it opens many possibilities. It kind of blows your mind, because the nervous system, unlike any other tissue, can plan ahead. It can synthesize information from the inside and also internal information, internal states, and plan and take many things into consideration. And if the nervous system can generate responses that will control the next generation, then it can, in theory, plan the fate or control the fate of the next generation. It sounds a little teleological, but it doesn't have to be. Okay, but it could be quite amazing. So what we did is we studied small RNA-based responses that originate in the nervous system. We used worms that make small RNAs just in their brains and studied the fate of these small RNAs, how they move between cells, and what happens if you just manipulate the biosynthesis of small RNAs specifically in the nervous system of the worm. And we saw that when you do this, you not only affect the worms that experience a disruption, but also you affect the progeny for multiple generations. And we found that worms that derive from ancestors that can't make small RNAs in their nervous system are different in many ways, including in their behaviors. Worms that derive from ancestors that 3 generations ago had their neuronal small RNAs disrupted have problems in finding food, in performing chemotaxis in slightly higher temperatures. And we identified the genes that are transgenerationally regulated by the nervous system and found one gene, it's called SAGE2, which is controlled by the brains of the ancestors, and is required, and this brain controlled transgenerational inheritance is required for proper chemotaxis. Okay. Now, I told you all of this introduction just so I can now move to tell you about a completely different story. Okay, it was a long way that we did to get to this point, but we had to do it because while we found that worms have very long-lasting memories that last across generations and last for months even, okay, When it comes to memories that are acquired within the same generation, that are kept in the nervous system, the worms in fact have a very short-term memory. Okay. So if you take a worm and you teach it, for example, you take a worm and you teach it that a certain odor that it likes, it's born liking this odor. If you teach it to dislike the odor by pairing it to starvation, you can grow the worms in starvation in the presence of this odor. Then the worms will learn to dislike the odor. However, these type of memories that are acquired within the same generation, are very short lasting. And after 2 hours, the worms will forget. They will again like the odor. They will forget that it was associated, that you associated it with starvation. And this is something that has been known for 30 years. People did memory experiments and found that they're very short lasting. And after I got you all excited about studying memory in C. elegans, you are probably a little disappointed because you say, well, they can learn, but they forget after 2 hours. So is it real memory? What can we learn about this type of memory? I think we can still learn a lot about this type of memory, but it is disappointing that it's so short lasting. Okay. The question is, why is it short? Why do they forget after 2 hours? So wait, there's a question in the questions. How do you assess if the worm likes or dislikes an odor? And that's a good question. So you can do it in a number of ways. The simplest way is you take a worm, in fact, you take multiple worms, you place them in the middle of a plate. In the one side of the plate, you put the odor that you're testing, and in the other side of the plate, you put a control odor. And you want to see whether the worms move towards the odor. If they move towards an odor, it means that they like him. They like this odor. If they do chemotaxis towards the odor, okay? And so in the example that I gave you, if you just take worms and test whether they like an odor like butanol or benzaldehyde, these are chemicals that the worms like, you will see that the worms prefer this odor over a control odor like ethanol or water. They will move towards the odor, and after an hour, you will find that all the worms in the plate reach the odor, reach butanol or benzaldehyde, these chemicals. However, if you take the worms and you grow them, you starve them, and you associate the starvation with butanol or with benzaldehyde, these odors, then you see that then when you test them, they don't go towards the odor. They stay in the middle of the plate, or they even go to the other side. Okay, this is how you test it. You can also look at the brain and actually see whether the neurons are responding. The worms are transparent, and we can assess we can measure neuronal firing and see whether they respond to an odor or not. But the simplest way is to just perform this chemotaxis assay on a simple plate. Okay, so you teach a worm something and it forgets after 2 hours. And then the question is, do they forget because they just have a bad memory? No, I mean, many times when we think about forgetting, you think of it as something bad. We want to remember better for an exam. Or we also know that when we are old or sick, then we forget. So we think forgetting is something bad. On the other hand, of course, forgetting is also adaptive. If we remembered everything that we ever learned, we would be completely post-traumatic. Also, we would remember many irrelevant information. So it's also adaptive to forget. The worms live for approximately 3 weeks in the lab. So still 2 hours is quite short, even for such a short lifespan. The question is, are they capable of holding memories longer in certain situations? Okay, now a short dramatic break while I'll drink this coffee. Okay, so I had an idea. I ended up never testing this idea, but this is how the project started. I wanted to teach the worm this association that an odor that they like is bad. And then I wanted to freeze them in the -80 refrigerator. You can freeze worms. We do it all the time. Okay, and then you can throw them after 10 years if you want. And I wanted to know whether the memory survives after freezing. That was the idea that I had. So I asked the student to do it. The student is Dana Landschaft, an excellent student in the lab. And she agreed to do it. But I found out that she didn't do it. Instead of doing what I asked her to do, she did a different experiment. She taught the worms this association. She grew them in low temperatures. She starved them in the presence of the odor to make them dislike the odor. But then instead of freezing them in the -80, she just grew them in low temperatures. She just placed the worms on ice, so about 0 degrees. And this is a low temperature, but the worms don't freeze. Okay, so they still have some electrical activity. Why did I want to freeze the worms? I wanted to see whether memory survives freezing, because if the memory survives freezing, it would mean that it can persist even when there's no electrical activity. And some of the theories about memory say that you need to have electrical activity to keep a memory going. And if the worms would still remember after you freeze them in the -80, it would mean that electricity is not important for maintenance of memory. But that never tested my idea, because she placed the worms on ice. And on ice, while it's cold, around 0 degrees, there's still electric activity probably. So it doesn't really test what I wanted to test. However, when she did this experiment that she invented, she found something very interesting. She found that for as long as the worms are kept on ice, they don't forget. Even 16 hours later, so 8-fold increase in the duration of the memory. You can even keep them for 24 hours and they will remember on ice. However, at this point, they become sick. So we do shorter experiments. But what she found is that as long as the worms are on ice, they don't forget. Memories are maintained on ice, which is very weird. We found that the ice doesn't consolidate the memory. It doesn't make it permanent, because as soon as you remove the worms from the ice, they start forgetting. And 2 hours later, they will forget. So it's just a temporary delaying of the forgetting that occurs on ice. We start thinking, what's going on here? Is it just a general slowing down of metabolism? Many reactions are slowed down in low temperatures, like the enzymatic reactions. Everything happens slower. Wait, there's a question in the question. Do you think this is due to some association with aversion? That's a complicated question. I will wait for it. Maybe we'll talk about it later. Okay, Shreya? Because I have a momentum here in the explanation, and I can go back to this later. So when we don't think, well, there's another question. If seeing has short-term memory, then will it be comparable with long-term memory by learning? So I'm waiting for a second with questions, because I want to tell you about the story, and then I will answer all these questions, okay? So great questions. I will go back to this. Okay. So we thought that perhaps memories are maintained on ice for longer, because in low temperatures, everything happens slower. So forgetting whatever it is slowed down. The breakdown of the memory, whatever it is, forgetting, just happens slower in lower temperatures. That was our original idea or thought, but we found that this is not true. I'll tell you why we think this is not true. There's been many studies on cold tolerance in C. elegans. If you take worms, and this has been studied for 20 years by other people. If you take worms and you put them on ice for 48 hours, they all die. However, it was found that if you take worms and you acclimate them gradually to lower temperatures, normally worms are grown in 20 degrees Celsius. If you acclimate them, if you get them used to growing in 15 degrees Celsius overnight, for example, you need a minimum of 5 hours, and then you place them on ice for 48 hours, they all survive. They don't die. And people studied what happened to the worms when you acclimate them to lower temperatures, and they found that they change lipid composition and all kinds of things that help them deal with the cold. Okay? So we wanted to see whether there's a link between cold tolerance and memory maintenance on ice. So we took worms, and before we taught them the association, we acclimated them to lower temperature. We grew them in 15 degrees Celsius instead of 20 degrees Celsius overnight. And then we taught them the association. We paired the odor with starvation. And we either placed the worms on ice or not. And we found that when you acclimate the worms, when the worms are made cold tolerant by acclimation to lower temperatures, then the memory is not extended on ice. Now, on ice, they forget after 2 hours. This means that the memory extension on ice is not because of physics. It's not because the temperature is lower and everything happens slower. It's because when they are on ice and they are not cold tolerant, when they are still sensitive, they change some internal state that supports memory maintenance. Okay? So we wanted to see what is this memory maintenance. So we took worms, we put them on ice, and we compared worms that are cold tolerant to worms that are cold sensitive. And we sequenced RNA from these worms. And we identified genes that are required for cold tolerance and also genes that are required for maintenance of memories on ice. For example, genes that are required for that participate in the production of diacylglycerol, if you disrupt this pathway, then the worms cannot maintain on ice. They are also incapable of becoming cold tolerant. And then we identified a gene, it's called TRX1, that if you, by sequencing, that if you, in TRX1 mutants, memories are extended, the worms do not forget after 2 hours, even when they are never on ice. Okay? This is very weird, and we are studying now the mechanism of this gene together with Antonio Miranda Vizuet, who studied the mechanism of TRX1 in C. elegans. We don't know what's going on there, and it's quite confusing. So I won't get into the details of this. I will just tell you that this gene is expressed just in a single neuron pair in C. elegans. These neurons are called ASJ. And the ASJ neurons were shown in the past to be required for cold tolerance. So we wanted to see whether the ASJ neurons can be manipulated to extend memories on ice. And we found in the literature that ASJ neurons are very special because these are the only neurons that respond to lithium. And lithium, some of you may know, the neuroscientists in the audience know, that lithium is a drug that is given to bipolar disorders for patients for many years. It's a miracle drug of psychiatry. And this is amazing because lithium is just an atom. It's the third atom in the periodic table, but it's been given as a drug for many, many years, although it's not entirely clear how it works. There's been a study that showed that when you expose one to lithium, the only neurons that are affected are the ASJ neurons, which are inhibited. And in fact, in that particular paper, they showed that when you expose worms to lithium overnight, the ASJ neurons stop firing, and also TRX1, this gene that we found to be associated with maintenance of memory on ice, is inhibited. So we grew worms on lithium and examined for how long do they remember, and we found that they don't forget. Unlike the wild type worms that forget after 2 hours, worms that are treated with lithium remember even after 5 hours, they still don't forget. And we are now trying to study the mechanism of how lithium extends memory, whether it's happening through the ASJ neurons, through TRX1, and how is it connected to low temperatures. We found that lithium-induced memory extension is affected by temperature. Okay, but the rest is still unknown, and we are still studying. This is all, results totally new that we are and still work in progress. But the bottom line is that C. elegans are capable of extending memories for longer, and we can study it, so we have a chance of actually understanding how memory is maintained. So to summarize everything, I will just tell you that worms are capable of transgenerational memories. They can have long-term memories in certain situations. And I think their simplicity makes them a very powerful model organism for understanding how the brain works and specifically how memory is encoded. And I will stop here. And of course, I'll be happy to answer any questions from the chat or also from whoever wants to speak. Thank you.
Thank you so much for this wonderful talk. I think there are quite a few questions in the chat box, and if you want to take them one by one, you can do that.
Oh.
Yeah, really, really fascinating talk. And I'm actually quite puzzled as well. I mean, it's so amazing to see molecular memories in something like C. elegans as well, but also because we talk about memory so much in terms of just the behavioral aspect and not being able to pinpoint molecules, basically. It's really, really fantastic to hear about something like this.
Thank you very much. Okay, so should I start? Where should I start? Okay, I will start in the question about how do we assess if this I answered. Do you think the memory is due to some association with starvation, with aversion? This is your question?
Yes, I think you already answered it in part, but if you want to expand, yes.
Okay, so I will continue to the following question. If C. elegans has short-term memory, then it will be comparable with long-term memory formation. Yeah. So the question is, if they only have short-term memory, what can we learn about long-term memories in humans? That is the question. Yeah. So as I've shown you, memories can be extended. So perhaps there's some similarity also with long-term memories in higher organisms. However, also short-term memory is very interesting. Why? And just recently, I think just yesterday or something, I saw an eLife paper about how short-term memory in crows shows the same characteristic as short-term memories in other organisms. So there's some universal similarities in the limitations of short-term memories, which I think are very relevant. You know, if you are given a few numbers to memorize, you have a very limited capability of memorizing them, and so on. And so I think even these type of memories that we consider short are fascinating to study. Okay, do fixed action patterns exist as molecular memory? I'm not sure I understand. Do you mean, Nilesh, if some instincts are encoded molecularly? If I understand it correctly, do I understand correctly the question? I'm not sure I understand. So perhaps later on, if someone wants to extend, I will be happy to answer.
Yeah, I think Nilesh, if you're around, you might as well like type your explanation if you want, or expand on your question.
Are there any other questions?
I think there was one more question by Keshav, which says, does age matter in memory formation and transfer? And are old worms bad at memory formation?
Yeah, so I can say a little bit about this first with regard to transgenerational memory. We studied it a little bit. There's been a paper, there's a paper in Nature from Ben Lerner's group. He showed that the descendants or the F1 project, this was just F1, just first generation, of old C. elegans mothers are different from progeny of young mothers. There's a difference there, and it's about allocation of yolk, provisioning with yolk, and the amount of yolk that the old mothers and the young mothers provide to their kids is different. So they are born a little different, more ready for life or less ready for life. When it comes to the transgenerational responses, we don't know enough. I do think that many things go wrong when the worms become very old. It's possible that the transgenerational small RNAs that the transgenerational small RNA responses that they initiate would be different, but this hasn't been studied extensively. When it comes to their capacity to learn just associations about the world, we didn't test it. I'm not sure if anyone did. Perhaps yes. We want to test the worms when they are in the best shape to learn something, because they're not that great at memory maintenance anyway. So we don't want to also complicate it with aging. But it's possible that someone has done it, and I'm pretty sure that if you try to teach very old worms something, that they will not acquire the memory so well. But I haven't tested it myself. So we now.
So there's a question from Sambit Dalui. It says, is memory through small RNAs transfer from one generation to another only through the ovum, or some paternal memory also transferred to the next generation?
Yeah, so this is another excellent question. Both the sperm and the egg can carry small RNAs transgenerationally. The worms are hermaphrodites, so they make both sperm and egg, but they're also males that just have sperm. Responses can also move through the males. Okay, the pool of heritable small RNAs in the sperm and in the egg is different, and also their capacity in terms of the intensity of the response is different. But both germ cell types can do it.
Oh, very fascinating. So another question from A. Karthik says, can the same effect as, I guess, with low temperatures be seen in high temperatures as well?
Yeah, we didn't study it. We did study a little bit whether the neuron that senses high temperatures affect the memory, and it does, but not as strongly. But we didn't study it yet. In very high temperatures, the worms are they will die. They can. Within the range, but we didn't compare the capacity to maintain memories, how long they maintain memories in slightly higher temperatures. This we didn't study, but we can definitely do it, and it's a good suggestion.
Okay, I think Nilesh has explained his question. He says fixed action patterns like the important behavioral patterns that are similar in all organisms of a single species.
Mm-hmm. So you mean?
I'm not sure what. Yeah,
I'm not sure. You mean what a very conserved response would be? I think perhaps you are referring to the conservation of these responses across different species. If that's what you're asking, so I'm not sure how to answer. I would just say that small RNA inheritance, or the capacity to inherit RNAi, is fastly evolving, and it's very different between different nematodes, even within C. elegans. Worms that are wild isolates that are collected in different parts of the world differ dramatically in their capacity to transmit responses transgenerationally. It's very fastly evolving. So, and this is partly because small RNA inheritance is a mechanism that likely evolved as a mechanism against viruses and transposons and things like this. So it's an arms race, which is, and the components are fastly evolving.
Cool.
Okay.
I think there are a few more questions, and in the interest of time, I think we will wrap up soon. So there's a question from Kazunori saying, I recall a talk by someone at an RNA splicing conference where he talked about lithium displacing magnesium in the RNA. Cannot recall if the lithium changed splicing pattern or not, but it's more a comment than a question. And if he can find this paper, he'll let you know, he says.
Yeah, it's an excellent comment. I would love to hear about it. If you do find the paper, please let me know.
And then there's another question from Rupkatha. She says, can the studies on molecular memories in worms give us an idea on tackling mental health issues in humans that arise as a result of traumatic memories?
So that's a $1 billion question. Is it conserved or not? The worms are capable of these amazing things, transgenerational memories. Do humans have it as well? The honest answer is we have no idea. It's very difficult to study because we are genetically different from one another. We have just a few kids. It's hard to do the statistics. The generation time is 20 years, so it's hard to study many generations. In C. elegans, the generation time is 3 days, and they make hundreds of babies. So you can study it very easily, and they're genetically identical. Only time will tell whether this is conserved or not. Currently, we just don't know, but I hope it is, but we don't know.
Right. There's another question from Sanjay Kumar Shukumar who says, how do small RNAs move from one cell to another? Do they have a cis-regulatory signal coded in the sequence? Can we block this kind of transfer?
Yeah, so most of what I can tell you is about double-strand RNA. Double-strand RNA, much easier to study because you can provide it from the outside, moves between cells in C. elegans, and there have been a few genetic screens on identifying components that are required for this non-cell autonomous response. Genes have been identified, for example, the SID1 transporter, which is, by the way, conserved across many organisms, which is just a transporter for double-strand RNA that physically shuttles the RNA across membranes. Okay, There are other components of a vesicular transport system that have been identified as crucial. We don't know, but all of these genes are required for import, not for export. How the RNA is exported out of the cell is not clear. It's possible that it's happening in some vesicles, but we don't know in C. elegans. And to tell you the truth, it's still a big mystery, and we don't know which type of small RNAs do it, whether they need to have some attributes, like certain modifications or certain structure or something like this. I just recently published a review about it last month about Nature reviews in molecular cell biology. So you can look it up. It's a very long review with everything that we know, but I admit we don't know enough.
I think in closing, since we really are running out of time, there are quite a few more questions, but I'll ask you two important questions about science as a research career, and what is your motivational 2 cents for all us young enthusiasts out here? If you can tell us something.
What is my motivation? What motivates me?
What motivates you, and what do you think should be our guiding light, sort of?
What motivates me is just to have an interesting life and to spend my days thinking about interesting things and being creative. That's my motivation. You know, of course, I hope it will be good or useful in some way, but even if it's not, and I just spend my day with creative and smart people thinking about interesting things, then I'm happy. I think it's a pretty good job. That's my motivation, to keep things as interesting as possible. And everyone has a different source of motivation. That's mine.
Right. Thank you so much for being here today. It was really a fantastic talk, and I think we all enjoyed it. Hope you have a very good holiday season as well. And thank you, the audience, for joining us for our last webinar, and we'll see you in the next year. And please keep watching us for updates coming up soon. Thank you all.
Thank you, everybody. Bye-bye.
Bye.